Okay, so right now we're talking about electron transport chain inside the final part of respiration. Okay, so respiration, and you're learning about ETC for the IB. So now let's start with the fundamentals. Where is respiration happening? So if I draw a cell, this is the nucleus. Okay, here I will draw a double membrane organelle. Okay, please excuse my fantastic art skills. Alright, so this is a mitochondria. You can see that I have drawn an outer membrane, an inner membrane, alright, in which the inside is called the intermembrane space. Okay? In between here, inside is the matrix. It's almost like the cytoplasm of a mitochondria. The ETC is located at the inner membrane. So I'm going to enlarge this portion. Whoop. This one line. This is the inner membrane of the mitochondria, this line. I will separate it into two areas. The top bit is the matrix, and this bit is the intermembrane space. Okay, now over here, I will draw a series of circles. Each one is an electron carrier. Basically what happens is this. In the whole process of the last stage of respiration, where the ETC is, you have a process called Oxidative Phosphorylation. Okay. Oxidation, that tells you there's a loss of electrons from a molecule. Phosphorylation, you are adding a phosphate to a molecule. So, as you have electrons, a pair of electrons being lost from a hydrogen carrier, which you produce from your glycolysis, your Lenz's reaction, your Krebs cycle, the electron carriers NADH will lose the electrons and put them into the electron transport chain. Okay, so NADH Whoop, becomes NAD plus. The electrons are lost. I'll tell you later where the hydrogen went. But the key is that as the electrons are moving in the ETC, you will have a pumping of hydrogen ions into the intermembrane space. So, I'll draw some arrows. And you see some H plus, H plus, H plus. You are building up a concentration of hydrogen ions inside this space. What would this do later? So, if I use a different shape, a triangle, this is a ATP synthase. Okay? As your electrons are pumping hydrogen ions, this ATP synthase are able to utilize this concentration of hydrogen ions to phosphorylate a molecule. So which molecule will you phosphorylate? In respiration, the molecule that you want to get energy, right? So the molecule that you want is ATP. Hence, this ATP synthase can get ATP, and you can add a phosphate to it, and you will get ATP. So, as your hydrogen ions are built up, it will go through the ATP synthase, and at the same time, this reaction is going to happen. Hence, you will see how oxidative phosphorylation, oxidation of the NADH, loss of electrons, at the same time, loss of hydrogen ions. The hydrogen ions can move from the matrix into the intermembrane space, And as the concentration build up, it can go back. It can go back to the matrix, but during this process, you will get ATP produced. This is why in your glycolysis, your Krebs cycle, your link reaction, you keep on producing NADH. You are getting reduced hydrogen carriers so that at the last stage, you will be able to convert this energy from oxidation into making ATP. Now, you will have a lot of ATP made using this process because, as you know in the IB, you have to distinguish aerobic respiration and anaerobic respiration. With each NAD and FAD that you oxidize, and you will make a lot of ATP. But if you're doing anaerobic respiration, you will limit it to your glycolysis, and then your pyruvate will be further becoming lactate, ethanol, CO2, depending if you're a plant or animal, right? You see that with this whole process, you will be able to unlock the energy inside the organic compounds, such as glucose, for ATP synthesis. And this is the core concept in the ETC. Thank you for watching. Remember to subscribe.